Electroporation can be done in vivo to transfect DNA plasmids and express transgenically in adult mouse flexor digitorum brevis, or FDB, and interosseous, or IO, muscles. The procedure begins with anesthetizing the mouse. The plantar surface of 1 foot is prepared for the first injection of hyaluronidase by inserting the needle at the heel and guiding it subcutaneously along the foot. After 1 hour, the animal is re-anesthetized and the plasmid is injected in the same subcutaneous region of the foot where the hyaluronidase was injected. After 10 minutes, gold-plated acupuncture needles are placed subcutaneously as shown. The electroporation pulses are delivered after connecting the needles to the stimulator. If needed, the procedure can be repeated in the contralateral foot. After a period ranging from 2 to 8 days, the animal is sacrificed, the muscles are dissected, and the transfection of the desired protein is verified by observing the fluorescence under a microscope.
Hi, I am Marino DiFranco from the laboratory of Julio Vergara in the Department of Physiology in the David Geffen School of Medicine at UCLA. Today we will show you a procedure for the in vivo transfection of transgenic protein-expressing plasmid into muscle fibers of adult mice. We use this procedure in our laboratory to study transgenic protein expression and localization, as well as the effect of the protein overexpression in muscle fiber physiology. So let's get started.
Before starting the in vivo electroporation protocols, mammalian expression plasmids must be amplified to yield